Hello, everyone. My name is Fabiha, and I will be talking about my master's applied project where I look at the effects of oxygen availability on neurodevelopment in organoid models. So cortical organoids are three-dimensional structures that are derived from stem cells, which are used to model brain disease, brain injury, and brain development. Essentially, these are little brain organs. So these organoid models are really helpful in looking at cell division and cell proliferation, as well as studying the features of endogenous tissue organization, and also looking at neurogenic to gliogenic transitions that happen in the brain. However, these organoid models are limited in their ability to reproduce complex brain physiology, refined cell types, and neural circuitry. So previous work in the lab has looked at organoid structures and noticed that these organoids tend to express high levels of cellular stress genes, which they noticed were reduced when these organoids were taken and transplanted into mice models. So it's unknown why this exactly happens, and is it likely that having a microenvironment that is similar to the in vivo brain, where these organoids are cultured in such an environment, might improve them as better models. So the goal of my project is to take cortical organoids and grow them for 5 weeks and under different oxygen conditions and see initial neurodevelopmental stages and see if there's an improvement when there's a lower oxygen condition that is more similar to the human brain. So the way I would do this is to grow induced pluripotent stem cells for 2 weeks and then dissociate them into single cells and aggregate them into organoids and place them into 96 well plates, where I will grow them for 35 days and then collect them, fix them, and then stain them to look at different cell types, as well as do qPCR analysis to look at some of the more genetic expression profiles. So the two ways I inhibited or reduced oxygen was in two different ways, where one way I inhibited reactive oxygen species. And when I did this, we noticed that there was when we inhibited reactive oxygen species, we noticed that there were strong morphological differences in the ROS1 organoid models, which were the ones that were introduced to this ROS inhibitor. And we found that there was a difference in size, shape, and density. However, these organoids were so fragile that we were not able to process them. Thus, we took a different approach in which we adjusted environmental conditions, which this was shown to be more reflective of the human brain in microenvironment. So when we lowered the oxygen conditions, we noticed that these organoids were having slightly higher diameters than the 20% oxygen conditions. And also we noticed that there was quicker development in terms of we found that Ki67, which is a marker for proliferating cells, went up, as well as CTIP2, which is a neuron marker. We noticed that that didn't change at all. However, we noticed that there was slightly higher cell death in the lower oxygen conditions than the higher oxygen conditions. However, we noticed because there was no change in the neuronal population and that they were roughly the same, there was no effect on the advancements of these lower oxygen condition organoids, and they were proliferating at a much faster rate and forming these rosette structures at a more faster rate than the control organoids. However, because it's unknown what cell types are actually vulnerable, for future works, we would try to identify which cells are actually vulnerable to cell death through secondary staining procedures, and also looking at single cell RNA sequencing to get some of that transcriptomic data if the secondary staining doesn't show us which cells are actually dying. Additionally, I hope to be completing a biological replicate with another cell line to make sure all these findings stand, and also possibly expanding the culturing time point to explore further neuronal development, as the timeline for the Master's Applied project made us have a shorter timeline versus having a longer one. We could look at some of those more advanced neurons. Thank you for listening, and I hope you come to my poster presentation. Thank you.